Hello, biochemistry students. Today I'm going to be doing a review that covers most of the major concepts of the material on exam 1. With that said, this is not a substitute for the lecture videos or class notes. Please use this as a review only, and as I go along, take note of the material that you feel unsure about, and be sure to refer back to your class notes and lecture videos for a deeper understanding. Non-covalent interactions are exceptionally important in biochemistry. The human body is a dynamic system. Bonds must constantly be broken and reformed during folding and unfolding of proteins. In order to keep up with all of the changes that occur, non-covalent interactions are energetically favorable over covalent bonds. There are 6 types of non-covalent interactions that we will focus on. You are expected to have a general idea of the strength of these. For starters, it is critical that you understand that covalent bonds are stronger than all non-covalent interactions. Pictured on the slide is a pyramid of the non-covalent interactions. At the bottom of the pyramid are the weakest: your hydrophobic interactions, pi stacking, and van der Waals. These all have a Gibbs free energy, or delta G, below 4 kilojoules per mole. Moving to the middle of the pyramid are electrostatic interactions and hydrogen bonds, which fall somewhere between 4 and 40 kilojoules per mole. At the top of the pyramid, we have the strongest non-covalent interactions, which are salt bridges, and they have a Gibbs free energy between 40 and 400 kilojoules per mole. Keep in mind that salt bridges are formed from a combination of hydrogen bonding and electrostatic interactions. As you can see, these two non-covalent interactions fall just below the salt bridges. Therefore, it makes sense that the combination of them would result in a very strong interaction. Water has a maximum of 4 hydrogen bonds per molecule. However, hydrogen bonding is only maximized during its solid phase, ice. On the other hand, as a liquid, it only averages 3.4 hydrogen bonds per molecule. With this information in mind, would ice or liquid or water have a higher volume? Ice would have the greater volume due to its maximum hydrogen bonding and lattice structure. So what about density? Remember, volume and density have an inverse relationship. Therefore, if ice has the higher volume, then it will have the lower density. So why is there a difference, you may ask? The difference lies in the fact that water does not exist as a single molecule. In liquid water, protons are constantly dissociating and reassociating. As soon as a hydrogen dissociates, it reassociates with a water molecule directly next to it, creating a chain reaction referred to as proton hopping. Realize that the hydrogen only hops to the water molecule directly next to it, and not any farther down the chain. In order to understand buffers, the Henderson-Hasselbalch equations, and isoelectric points, you must first have a basic understanding of acid and base measurements and symbols. These are things that you learned in general chemistry, but may have forgotten, so we're going to go over it. The brackets are symbolic of concentration. When you see HA in the brackets, it is a generic form of an acid. And the A- in the brackets, or the A negative, is symbolic of a base, or the conjugate base. The higher the concentration of HA in comparison to the A-, the more acidic the solution is. The opposite is true for a base. The greater the concentration of the A- or the base, the more basic the solution. Now is a good time to review pKa and Ka. Keep in mind that they have an inverse relationship. The higher the Ka and the lower the pKa, the stronger the acid. The Henderson-Hasselbalch equation is often used when estimating the pH of a buffer system, and we use it to compare acids and bases, or acid and conjugate base concentrations. As I stated here in the slide, it is not just about being able to calculate it. You probably all did that in Gen Chem already. In biochemistry, we expect that you are able to take the calculation and apply the knowledge. At the bottom of your slide is the way that I like to think about it. So let's pretend that you already did your calculation and your base over your acid comes out to a value of 1, or your A over your HA is a value of 1. Well, if your pH is equal to the given pKa plus 1, then your conjugate base is 10 times more concentrated than your acid, or your HA. This makes sense because if you have more base than acid, your pH will be higher. Just as a side note, please remember that one pH unit has a 10-fold change in hydrogen concentration. So if your pH is increased by 1, it's 10, but if your pH is increased by 2, that would be 100. The same goes for the example below. Only now, we have a lower pH than pKa. Thus, our acid will be more concentrated than our base. I encourage that you spend a little time rationalizing this out. It will truly help you when solving Henderson-Hasselbalch problems if you can tell whether or not your answers make sense, and not simply rely on a number that you inputted into your calculator. Pictured on this slide is the generic structure of a nucleotide. When we talk about the DNA or RNA bases, adenine, guanine, cytosine, thymine, and uracil, we are talking about nucleotides. However, there is more to a nucleotide than a purine or a pyrimidine base. A nucleotide consists of 3 main parts, which are listed here. A pentose sugar, either a ribose or a deoxyribose, connected to a phosphate group at its 5' end over here, and a purine or pyrimidine base, which you can see right here, connected to the carbon 1 of the sugar. In RNA, we see a ribose sugar, which has a hydroxyl or an OH group, pictured right here in red. However, in deoxyribose, the hydroxyl group is replaced by a hydrogen. Chargaff's rules allow us to determine the percentage of DNA bases if given the percentage of one of the 4 base pairs. For example, if you are given the percentage of adenine, you are expected to be able to determine the amount of not only thymine, but also guanine and cytosine. The percentage of adenine will equal the percentage of thymine because they are base pairs. Similarly, the percentage of guanine will be equivalent to the percentage of cytosine. Let's try out the example on this slide. The question states that a given sequence contains 30% guanine. Using Chargaff's rules, we are going to need to figure out the percentage of cytosine, adenine, and thymine. Take a minute to try this out. All right, well, let's solve it together and see how you did. First, let's think of our base pairs. So what pairs with guanine? Cytosine. Therefore, their percentage composition must be equivalent. So if we have 30% guanine, we must also have 30% cytosine. So how much is left over? Well, we have 60% accounted for, 30% C and 30% G. So if we take 100% and we minus the 60%, we are left with 40%. Because adenine and thymine are base pairs, Chargaff's rules tell us that they must be equal. So we take the 40% and divide by 2. That gives us 20% for each. When checking your answers, always make sure that the 4 base pairs add up to 100%. And remember that uracil, which is an RNA, is never an option. Chargaff's rules only apply to DNA. Another important fact to remember about base pairs is the amount of hydrogen bonding. So adenine and thymine have 2 hydrogen bonds per pair, whereas guanine and cytosine have 3. The increase in hydrogen bonding results in greater stability, which is what leads to G and C having a higher melting or denaturation temperature than AT. The graph on your right displays just that. Amino acids are at the heart of biochemistry. Being able to identify them by their structure, their 1- and 3-letter codes, and their properties is essential for not only this exam, but for all of your upcoming exams as well. I'm not going to go over them today, but I suggest that you write them out on your own and categorize them by their properties if you haven't already. Every amino acid has an amino and a carboxyl terminus, referred to as the N-terminus and the C-terminus respectively. The side chain is what makes each amino acid unique and what is essential to understand when solving isoelectric point problems. When determining an isoelectric point, keep in mind that not all amino acids have ionizable side chains. For example, lysine is commonly seen in isoelectric problems, and it has an ionizable side chain. However, glycine does not. Therefore, glycine does not. Therefore, lysine will have 3 pKa values, whereas glycine will have only 2. Recognizing what amino acids have ionizable side chains will speed up these problems and allow you to solve them much more efficiently. With that said, remember that if on either the N-terminus or the C-terminus of a peptide sequence, every amino acid will be ionizable. In other words, glycine is not ionizable if it's in the middle of a peptide sequence, but if it's the first amino acid of the given sequence, its N-terminus will not be participating in a peptide bond, so it will be able to be deprotonated. Another topic I want to mention is titration curves. A longer vertical line, as you can see right here, where the pi equals 5.97, is representative of the isoelectric point. An isoelectric point is reached when the amount of deprotonated species is equal to the protonated species, making the overall net charge neutral. Now notice the nearly horizontal portions of the graph. These are surrounded by the blue boxes right here and up here, where you see the pK1 and the pK2. These are indicative of the buffering region, which is optimal at 1 pH above or 1 pH below the pKa. compounds are capable of absorbing ultraviolet light. The amount of absorbance is proportional to their size. The larger the aromatic compound, the greater its absorbance. If you know your amino acids, this makes remembering their absorbency relatively easy. Phenylalanine is the smallest, therefore it will absorb the least, whereas tryptophan is the largest and is capable of the greatest absorption. Note that proline is not a key player in UV light absorption and can be easily thrown into answer choices to trick you. Proline is cyclic, however, it is not aromatic. Remember, aromaticity involves conjugation. This slide is just a reminder of the nomenclature and molecular weight of amino acids. When amino acids come together and form a peptide bond, they are called residues. Once part of a peptide chain, all amino acids, but the last, or the C terminus, of an amino acid will end in YL. Down at the bottom of the slide, you see the average molecular weight of an amino acid residue. You should be able to determine the number of amino acids in a peptide sequence simply by knowing their weight. If you are given a value, let's say 2,000 Daltons or grams per mole, you divide that number by 110. For this example, you would have about 18 amino acid. Realize that this is just an approximation, and you may not end up with a perfectly even value. Don't be concerned. Peptide bonds have double bond character that restrict them to the trans conformation, which has a bond angle of 180 degrees. This can be seen by the resonance structure displayed on the slide. The nitrogen donates its lone pair to the carbon-nitrogen bond, causing the pi bond of the carboxyl group to break. You can see here, the nitrogen donates its pair. Now carbon has too many bonds, therefore it breaks its pi bond and donates its electrons up to the oxygen. The newly created pi bond of omega now restricts its movement. Keep in mind that proline is an exception, and it typically has a cis conformation. There are 4 structural levels that you need to be familiar with. Understanding both the structures and the types of bonding involved is crucial. The primary structure is simply the amino acid sequence. At this early stage, peptide bonds, which are a form of covalent bonding, are essential. Moving on to the secondary structure, we become concerned with hydrogen bonds. There are 2 main structures to take note of when discussing secondary structures: alpha helices and beta sheets. We will discuss more on them in an upcoming slide. Tertiary structures mark the beginning of the 3-dimensional fold that is required for biological activity. Hydrophobic interactions are key players in determining a protein's tertiary structure. Unlike the first 3 levels of structures that focus on a single polypeptide chain, quaternary structure is only reached if 2 or more polypeptide subunits associate. As a side note, in order to have cooperativity, a protein must have quaternary structure. While we separate each structural level for educational purposes, please do not think that polypeptides follow a sequential order. As the polypeptide grows, all levels, except for quaternary, which may never be reached, are occurring at different places at different points in time. Therefore, some parts of the amino acid may be at secondary structure, some may be at tertiary, and so forth. The hydrogen bonds in both alpha helices and beta sheets are between main chain atoms, specifically between the carbonyl oxygen and the hydrogen of the amino group. On the far left, you can see an alpha helix, which is right here. Here's your alpha helix. There are approximately 3.6 residues per turn, and a regular repeat leads to this alpha helix. A few things to keep in mind when answering questions regarding alpha helix is that proline breaks the helix and that glycine is very rare. Looking at the right side of your slide, you see two different beta sheets, parallel and antiparallel. Because linear hydrogen bonds have the greatest stability, antiparallel beta sheets are more stable than parallel beta sheets. They have the maximum number of hydrogen bonds, and therefore, the maximum number of water molecules are released. Remember that for each hydrogen bond, one molecule of water will be released. Ramachandran's plots display the bond angles allowed in secondary protein structures, specifically phi and psi, because omega has minimal rotation due to its double bond character. While you do not need to be able to draw a structure simply by looking at this plot, it is important that you are able to recognize what type of secondary structure falls into each area. The red regions display the least steric hindrance, thus their optimal bond angle. Please take note of where the different secondary structures fall on the plot, such as the upper left-hand corner where we see the beta sheets, and down below where we see the right-handed alpha helix. Hemoglobin and myoglobin share several similarities, but also some significant differences. Due to time constraints, I'm not going to go over each similarity and difference listed. Many of these points will be reiterated in the next few slides. But I suggest after this review, you go back to this Venn diagram and review the major differences. However, before we move on, I want to point out a couple of things just to make sure that you have a basic understanding of the differences between myoglobin and hemoglobin before we begin looking at their saturation curves. I like to think of hemoglobin as transporters because red blood cells use hemoglobin to transport oxygen from the lungs to the tissues. Additionally, hemoglobin is also used to return carbon dioxide to the lungs in order to be exhaled. Think of hemoglobin like a taxi. It is used to get from point A to point B. Conversely, myoglobin binds and stores oxygen. Because it is involved in oxygen storing, it must take the oxygen from hemoglobin. The only way for it to obtain oxygen from hemoglobin is if it has a higher affinity for it, or for the oxygen, than hemoglobin. Adult hemoglobin which is the hemoglobin that we will be dealing with this class, is referred to as just HB. And hemoglobin, in its tetrameric form, is A2B, alpha 2 beta 2. Only as a tetramer does it have quaternary structure and thus displays cooperativity. The saturation curve on your left displays this. Notice how it is sigmoidal or an S shape, see right here? Keep in mind that hemoglobin, although it does display cooperativity, it does not display perfect cooperativity. It has 4 possible binding sites. So according to Hill's coefficient, a value of 4 would be perfect. Hemoglobin displays about 3. Now, let's take a look at the saturation curve on the right. Hemoglobin subunits have now been dissociated by mercury 2 chloride. So we have the alpha subunit and the beta subunits by themselves. Because the subunits are no longer interacting with one another, it does not display cooperativity. Notice the difference in the shape of the curve. It is no longer sigmoidal or S-shaped. It is now hyperbolic. See how it reaches saturation at a much lower partial pressure? Here we are comparing myoglobin to hemoglobin in its dissociated or non-tetrameric form. As you can see, they display very similar curves. Both hemoglobin and myoglobin titrate very early. They are both hyperbolic. With this in mind, does myoglobin display cooperativity? No, it does not. What about a quaternary structure? Does myoglobin obtain the structural level? If you said no, you are correct. Myoglobin is a monomer. Therefore, it does not display cooperativity. Let's think for a moment. What problems could occur between myoglobin and hemoglobin if hemoglobin is not displaying cooperativity? How does this change the KD, or the affinity, make a difference? Remember from a few slides ago that we discussed how hemoglobin is the transporter, or the taxi, and myoglobin is the one that binds and stores oxygen. Well, in order to obtain the oxygen from hemoglobin, we said that myoglobin must have the higher affinity. If hemoglobin is not displaying cooperativity, myoglobin is going to have an increasingly difficult time binding and storing this oxygen. Making these connections will allow you to retain this information more easily and apply it to questions that may be a bit outside the box. Two,3-bisphosphoglycerate is an adaptive mechanism that becomes increasingly important at higher altitudes because less oxygen is available. Under these conditions, 2,3-bisphosphoglycerate will be synthesized, which enables more oxygen to be released in peripheral tissues. If looking at this on a dissociation curve, you will notice a shift to the right. This will be similar to what we see called the Bohr's effect. So up top here, you can see a little flow chart that I made. We have, at a higher altitude, we'll have less oxygen available. So our 2,3-BPG synthesis will be induced, and more oxygen will be released to our peripheral tissues. However, keep in mind that when returning to lower altitudes, there will be a decrease in pH. And when there's a decrease in pH, 2,3-bisphosphoglycerate synthesis will be inhibited. Before we move on to enzyme kinetics, I want to take a brief moment to review thermodynamics. The equation governing most of the thermodynamics of this course will be the Gibbs free energy equation, which is, as you can see on screen, delta G equals delta H minus T. There's actually a typo on screen. It's supposed to be delta H, or delta G equals delta H minus T delta S. Please make changes to your notes on this. It is important that you understand the different components of this equation and do not mix them up, particularly enthalpy versus entropy. Enthalpy, which is delta H, is the total heat content of the system. When you see the H, think heat. Entropy, on the other hand, is the randomness of the system. It is denoted as delta S. The goal of most systems is to increase their randomness. Thus, a positive delta S value is optimal. So what about the free energy of the equation? The delta G. What is considered favorable? Positive or negative? Well, unlike delta S or the entropy, where positive is optimal, a favorable reaction will have a negative delta G. A negative delta G indicates a spontaneous reaction. There is one last point I want to make. Enzymes do not affect the thermodynamics of a reaction, only the kinetics, or in other hands, the rate. As we move forward in our discussion on enzymes, I will reiterate this point so that you keep it in mind as you go throughout this course. As you can probably already tell, biochemistry is all about enzymes. They are everywhere and they will occur in every exam's material. Having a solid foundation right from the start will make not only this exam's material, but also future material in this course easier to understand. Before we go into the different types of inhibition and more detailed specifics, let's first review what exactly an enzyme does. Well, we all probably already know that enzymes are catalysts and they increase the rate of the reaction. But how does an enzyme do this? What does it affect? As we stated on the previous slide, enzymes affect the kinetics of a reaction, and they do this by lowering the activation energy, typically written as E sub A. Basically, it causes the reaction to speed up. Don't be fooled though. It does not allow a reaction that wouldn't already occur to happen. Therefore, it does not affect the thermodynamics, such as the equilibrium or Gibbs free energy. If a reaction is non-spontaneous, adding an enzyme will not make it spontaneous or not make it occur. Another important fact to keep in mind about enzymes is that many of them contain cofactors or coenzymes. Cofactors are typically metal ions, and coenzymes are usually vitamins. You will notice this trend when we begin learning about cellular respiration in the next exams material. But for now, keep this fact in mind. Furthermore, enzymes are never consumed in a reaction. This is why when writing a balanced equation in general chemistry, or even when we write for, like, let's say for glucose or glycolysis, the enzymes are not included. It does not mean that they weren't present. It's just that since they were regenerated in the same form that they began, they were not included. They canceled out on both sides. This slide is a visual representation of what I just explained. I'm not going to sit here and spend any more of your time explaining how enzymes work, but I wanted to include this for those who are more visual learners. Enzymes are specific. They have unique interactions between the amino acid side chains that work together to create a unique microenvironment for optimal chemistry to occur. The image on this slide shows that different enzyme configurations. Take a look at the changes in the red and blue pockets. Which configuration do you think is more favorable? The L-alanine on your left or the D-alanine on your right? If you chose L-alanine, you are correct. Good work. So, why did you choose it? Well, if you said it's because opposites attract, then you're right again. On the left of L-alanine, we see a carboxylate group next to a positive charge. Right here, you have your carboxylate group and your positive charges. At the top, there is a group of negative charges next to a positively charged amino group. And finally, at the bottom, we see a methyl group in a hydrophobic pocket. Everything here agrees. However, on your right with D-alanine, we see a negative charge here in the hydrophobic area, or the hydrophobic pocket. Remember, negative charges typically are polar, and hydrophobic pockets are nonpolar. They don't want to be together. Two different models have been proposed to explain how enzymes and substrates bind. On the left, there is the lock-in key model, and on the right, the induced fit. In the lock-in key model, the enzyme is complementary to the substrate. Notice how low of energy the enzyme-substrate complex, labeled ES right here as you can see, is. Because the free energy is so negative, the distance to the transition state is exceedingly high. Your transition state is right up here, marked in the green with the double daggers. Basically, the enzyme is so happy that it is highly unlikely that it would ever reach the transition state. This is why this is not regarded as the most favorable model to explain enzyme and substrate binding. Now, let's take a look at the induced fit model here on your right. where does the enzyme substrate complex fall in regards to the free energy? We see that it is not any lower than the substrate alone, and it is also higher than the product. This is because the induced fit model proposes that the enzyme is not complementary to the substrate, but it is instead complementary to the transition state. The enzyme binds the transition state with a higher affinity than the substrate or the product, resulting in equivalent rates for both the forward and the reverse direction. So keep in mind, in the lock and key model, which is not favorable, the enzyme is going to be complementary to the substrate, and in the induced fit model, which is the favorable one or the one that you want to keep in mind when you're thinking about how enzymes and substrates interact, the enzyme is going to be complementary to the transition state, not the substrate. Nevertheless, when you think of catalysis occurring, just always keep in mind the induced fit model. For your exam, you may be asked to identify the different types of inhibitors based on a Lineweaver-Burke plot. Therefore, it is important that you understand not only how the different inhibitors affect the Vmax and the Km, but also how to interpret them on a graph like you see on the slide. So pictured on the left, we have the Lineweaver-Burke plot of a competitive inhibitor. These reversible inhibitors bind at the active site and can be reversed by increasing the substrate concentration. Because they are capable of being reversed, the Vmax does not change. What competitive inhibitors affect is the Km, or the affinity. Recall that the lower the Km, the higher the affinity. With this knowledge in mind, how do you think a competitive inhibitor affects the Km? Does it increase or decrease it? There are two ways to think of it. A competitive inhibitor decreases the affinity. However, remember that the lower the value of Km, the higher the affinity. So what we're going to see is an increase in the value, or a higher number for Km. Take a look at the blue versus the purple line on your left. The purple line displays the enzyme kinetics prior to adding the competitive inhibitor. Comparing the two, we see that the blue line is more positive. Thus, it moves to the right on the x-axis, indicating an increase in Km. However, the y-axis, which is where we find our Vmax, is the same, which is right here. So the blue line, which is your competitive inhibitor, moves to the right, which says that we're going to have a lower affinity for Km, or a higher value, and our Vmax is going to stay the same. Now let's look to your right at the uncompetitive inhibitor. What makes an uncompetitive inhibitor uncompetitive is that it binds at an allosteric site instead of an active site. By looking at the Lineweaver-Burke plot, we see that both the Km and the Vmax change. Keep in mind that Lineweaver-Burke plots are double reciprocal plots. Thus, the higher up on the y-axis the line falls, up here, the lower the Vmax will be, opposite of what you might think. Take a look for a minute at the Km. Notice how it moves to the left, right here? Didn't we determine that moving to the left means an increase in affinity? We did, but believe it or not, an uncompetitive inhibitor actually decreases the value of Km. When the inhibitor binds to the allosteric site, it literally locks in the substrate to the active site, preventing its release. To sum it up, when you see an uncompetitive inhibitor, expect to see parallel lines with a decrease in both the Vmax and the value for Km. The other type of inhibition we need to go over is the mixed inhibitor. This one has a few tricks up its sleeve and can be a bit harder to identify. But keep in mind that a mixed inhibitor will always decrease the Vmax. However, it can either increase or decrease the Km depending on its specified type. Fatty acids are carboxylic acids with hydrocarbon tails. Their oxidation is highly exergonic, and as a result, a lot of energy is released. Our bodies produce even number chains, and their nomenclature indicates both the length of the chain and the number of double bonds. Saturated and unsaturated fatty acids have some different important properties for you to consider. Let's start with the saturated fatty acids. Because they do not contain any double bonds, they have the maximum number of hydrogen bonds, hence the name saturated. And therefore, they are free to rotate. They also have a higher melting point. An unsaturated fatty acid is a fatty acid that contains one or more double bonds. In physiological conditions, we find these bonds in the cis conformation. Because of the pi bonds, they are restricted in their rotations, and they have a much lower melting point. Think of olive oil full of unsaturated fatty acids versus butter. Last but not least, I want to review the two main types of lipids you will need to know. First, we have the energy storage lipids, such as fatty acids, triglycerides, and waxes. Second, we have the structural lipids that include all of the membrane structures you see listed on the right. The structural lipids are important for both membrane structures and cell surface sites of biological recognition. The structural lipids for you to keep in mind are your glycerophospholipids, your sphingolipids, your ether-linked lipids, and your sterols, which we often discuss as cholesterol. I'm not going to sit here and go over them individually with you today, but I suggest that before your exam, you review the main difference amongst each of them, especially the different types of bonds and head groups, as well as review the different chromatography techniques that I have provided on the following slide. Good luck in your studying, and if you have any questions on the material, please contact your TA. Thanks.